Welcome back to the deep dive. Today, I want you to picture a map. Not a map of a city or a subway system or even the stars. I want you to picture a map of life itself.
Okay.
If you zoom in on a single human cell and you try to trace where all the energy comes from, you see pathways just sprawling everywhere. You see lines for glycolysis, lines for fatty acid breakdown, lines for amino acids. It honestly looks like a chaotic, tangled mess.
It does. It looks like spaghetti junction.
But if you trace all those lines, if you follow them inward, every single one of them, every one, eventually converges on one central hub.
The grand central station of the cell.
Exactly. We are not hanging out in the metabolic suburbs today. We're going right to the absolute center of the map. We are covering the topic simply titled the citric acid cycle.
Or the Krebs cycle, or the TCA cycle for tricarboxylic acid. It goes by a lot of names, but the function is singular. It really is the metabolic crossroads where everything converges.
And for this investigation, we're synthesizing two, absolute titans of the biochemistry literature. We've got Leninger's Principles of Biochemistry, which gives us that beautiful architectural systems view, the chemical logic of why things happen.
The why. Yeah.
And we have Harper's Illustrated Biochemistry, which is going to keep our feet on the ground in the gritty clinical reality of, what happens when things go wrong?
And that combination is just so crucial for this topic. Leninger tells you how the engine is built, every nut and bolt, but Harper tells you why the car stops running and, more importantly, how you might go about fixing it. It's the perfect pairing.
But before we dive in, I want to be really clear with you listening. This is our interpretation. We're synthesizing thousands of pages of very dense science into a conversation. We want you to use this as a map, but you still have to walk the territory yourself.
Please do.
Open the books. Look at the structures, the diagrams. We're the tour guides. But the landscape itself, that's in those texts.
Absolutely. The goal here is to give you those aha moments, the little clicks of understanding that make reading the chapters feel like you're just filling in the details rather than, hacking your way through a dense jungle.
So let's set the stage. We're going to adopt a persona today. We're not just podcast hosts. We are Medlock Holmes, metabolic detectives.
I like it.
We've shrunk down to the size of a proton, and we are stepping inside the mitochondria to observe the architecture of energy itself.
And as detectives, we're investigating a mystery. And the first mystery is really a design question, isn't it?
It is. This is the why question that always bothered me back in high school biology. We have this fuel molecule, acetyl-CoA. It's a tiny 2-carbon thing. We want to get energy out of it. Why don't we just burn it?
Right. Just oxidize it directly.
Yeah. Why do we need this massive, complicated 8-step merry-go-round just to break a couple of chemical bonds? It just feels so over-engineered.
It does look like a Rube Goldberg machine at first glance, I'll give you that. But think about the alternative for a second. If you took acetyl-CoA and just oxidized it all at once, essentially burning it, you would release a massive amount of free energy instantaneously.
Like a tiny explosion.
Exactly. It would be a bomb, not an engine. The heat released would be so intense, the cell would physically cook itself from the inside out. The proteins would denature. The genius of the cycle, and this is something Leninger emphasizes so well, is that it is a controlled burn.
Okay, controlled burn.
It fractures that huge energy release into 8 smaller, much more manageable packets. It lets the cell capture that energy methodically in the form of these electron carriers, NADH and FADH2, instead of just losing it all as useless and, frankly, dangerous heat.
So it's really the difference between tossing a tank of gasoline onto a bonfire versus running it through the fuel injectors in a high-performance car engine.
That is a perfect analogy. But there's a second reason, and you could argue it's even more important for explaining the complexity. If you just burned the fuel in that bonfire, all you'd have at the end is CO2 and ash. But the cell isn't just a power plant, it's also a factory. It needs raw materials. It needs building blocks.
This is the versatility argument. This is the other half of the story.
Precisely. By spreading the oxidation process over 8 distinct steps, the cycle creates this whole series of strange, half-finished intermediate molecules. And it turns out those intermediates are the Lego bricks for the rest of the body.
What do you mean by Lego bricks?
Well, they're the starting points for making amino acids, for synthesizing glucose when your blood sugar is low, for making the heme group in your hemoglobin. If you just burn the fuel instantly, you wouldn't have any of those vital building blocks available.
So the cycle is really doing double duty. It's a power plant generating ATP, but it's also a parts factory, churning out supplies for other projects.
And that is why it's the Grand Central Station. It's not just about trains passing through. It's about passengers getting off, changing lines, and heading off in completely new directions.
Okay, Medlock Holmes is ready. The investigation begins. So let's start at the entrance. We're standing just outside the cycle proper. We've got our raw material, pyruvate, which has just arrived from glycolysis. We are ready to enter the factory floor. But we can't just walk in, can we? There's a barrier.
No, there's a gatekeeper, a very important one. Pyruvate is a 3-carbon molecule. The cycle, the main engine, runs on 2 carbon units of fuel. So first, we need to do a bit of prep work. We need to strip 1 carbon off and attach the remaining 2 carbon piece to a special carrier molecule.
And this all happens in a truly massive molecular machine called the pyruvate dehydrogenase complex, or PDH for short.
And when you say massive, I feel like words kind of fail us here.
Yeah.
Leninger says this complex can be up to 10 million Daltons in size.
10 million. Just to put that in perspective for you, what does that mean?
Our ribosome, which we usually think of as a pretty big piece of cellular machinery, is tiny compared to this thing. The PDH complex is larger than a ribosome. If you could see it under an electron microscope, it doesn't look like a typical protein blob. It looks more like a raspberry or maybe a soccer ball that's just bristling with little arms.
Molecular soccer ball. And it's not just one enzyme doing one job, right? It's a whole cluster.
It's a beautifully organized, self-contained cluster of 3 different enzymes, and there are dozens of copies of each one. We just call them E1, E2, and E3 for simplicity. The E2 units make up the inner core, that's the soccer ball shape, and then the E1 and E3 units are wrapped around the outside, kind of like a shell.
Okay, and this structure, this architecture, leads to one of the most fascinating mechanical concepts in all of biochemistry, substrate channeling. This solves a huge logistical problem for the cell.
It solves the diffusion problem.
Right. Explain that to me, because my default mental image is just chemicals floating around in a kind of cellular soup until they randomly bump into the right enzyme.
And, for some simple reactions, that works just fine. But if you have a multi-step process like this one, diffusion is incredibly slow and inefficient. Imagine enzyme A does its job and then releases its product. That product then has to just wander aimlessly through the incredibly crowded mitochondrial matrix to find enzyme B.
It wastes time.
A huge amount of time. And it's risky too, right? Leninger makes a big point about side reactions.
They're kind of risk.
It's like a relay race where after the first leg, you just drop the baton on the ground and hope the next runner finds it somewhere in the grass. Someone else might pick it up by mistake. A side reaction could happen, stealing your intermediate. So the PDH complex solves this by essentially cheating physics. It uses a physical tether.
The swinging arm?
The lipolysyl arm. It's a long, flexible arm that's chemically attached to that E2 core. The best way to think of it is like a robotic arm on a car assembly line. It is long, flexible, and it's bolted to the center of the machine.
So walk me through the movement. What is it actually doing?
Okay, so the arm swings over to an E1 enzyme where the pyruvate is being decarboxylated. That's the CO2 getting clipped off. The arm then picks up the remaining 2-carbon acetyl group. It literally grabs it.
Okay.
Then the arm physically swings that intermediate across a gap to the active site of its own E2 core. It processes it, hands the acetyl group off to its final carrier, coenzyme A, and then the now empty arm swings over to an E3 enzyme to get reset, to get reoxidized for the next round.
So the intermediate molecule, the thing being worked on, it never actually leaves the surface of the complex.
Never, not for a nanosecond.
Yeah.
It is hand-delivered from one active site to the next. It's like a relay race where the runners are bolted to the floor and they just pass the baton hand to hand. The efficiency gain is just massive. No lost time, no stolen intermediates.
That is just incredible molecular engineering. But as Medlock Holmes, I have to look for the vulnerabilities. A machine this complex, this precise, must have a breaking point. And Harper gives us a pretty chilling list of ways this gateway can be slammed shut.
And this brings us to what I call the famous 5 cofactors. The whole PDH reaction requires 5 specific helper molecules to work. You need thiamine pyrophosphate, FAD, coenzyme A, NAD, and lipode.
And 4 of those are derived from vitamins.
Right. Thiamine is vitamin B1. The FAD comes from riboflavin, which is B2. NAD from niacin, B3. Coenzyme A from pantothenate, B5. This is the direct, unbraidable link between your diet and your ability to generate energy. If you don't eat these vitamins, the PDH machine literally lacks the parts it needs to run.
Okay, let's focus on one of those. Thiamine, vitamin B1. A deficiency here leads to a condition called beriberi. And Harper points out something really interesting. Beriberi affects the brain more severely than almost any other organ. Why is that?
It all comes down to fuel preference. Your muscles, your heart, your liver, they're metabolically flexible. If the PDH complex is broken and they can't burn sugar efficiently, they can just switch gears and start burning fatty acids instead. They have a backup generator.
But the brain is a picky eater.
The brain is an absolute energy hog, and it relies almost exclusively on the aerobic oxidation of glucose. It needs that steady, uninterrupted stream of acetyl-CoA coming from pyruvate. So if you have a thiamine deficiency, the E1 part of the complex breaks down, the gateway is locked shut, the brain literally begins to starve, even if the rest of the body has plenty of fuel available.
And historically, this was a huge problem in populations that relied on polished white rice, where the vitamin-rich outer husk was removed. But Harper brings it right into the modern ER. We see this today in cases of severe alcoholism.
We call it the empty calorie problem. Alcohol itself contains plenty of energy, lots of calories, but it has zero vitamins, absolutely none. So if you're an alcoholic and you're getting a significant portion of your daily calories from distilled spirits, you're fueling the body, but you're not providing any of the tools needed to process that fuel or any other fuel. You eventually just deplete your body's thiamine stores.
So you essentially give yourself nutritional beriberi.
You do. Clinically, it presents as Wernicke-Korsakoff syndrome. You see confusion, problems with walking and coordination, profound memory loss. It is a metabolic tragedy caused by a broken gateway enzyme.
But there's another villain in this story, and it's not a deficiency, it's poison. I was reading about arsenic and mercury. This connects back to that robotic arm, right?
This is pure, brutal chemistry. Remember I said that swinging arm, the Lapoda arm, picks up the acetyl group? To do that, the very tip of the arm has two sulfur atoms, we call them thiol groups, positioned right next to each other. It's like a tiny two-pronged claw.
And heavy metals like arsenic, they just love sulfur.
They have an incredibly high affinity for it. It's one of the strongest chemical attractions. So if arsenic gets into your mitochondria, it sees that two-pronged claw, and it binds tightly to both sulfur atoms simultaneously.
It handcuffs the robot arm.
That is exactly what it does. It forms a stable ring and locks the arm in place. The arm can't swing, it can't pick up fuel, it can't pass it on. The entire PDH complex jams solid. Energy production grinds to a halt. That is why arsenic is so deadly. It is a molecular monkey wrench thrown directly into the gears of one of the most important machines in the cell.
Okay, that is a terrifyingly precise image. But let's assume we've eaten our vitamins and we've avoided any arsenic-laced tea. The PDH complex does its job. It converts our 3-carbon pyruvate into a 2-carbon acetyl-CoA. Now, finally, we're ready to actually enter the cycle.
We're at the starting line, step 1.
And immediately, Leninger highlights another chemical problem. We've got our acetyl-CoA. It has 2 carbons. One is a reactive carbonyl group, but the other is a methyl group, a CH3. And apparently, methyl groups are chemically very stubborn. They just don't want to react.
They're very stable, very unreactive. Oxidizing a methyl group directly under the mild conditions inside a cell, neutral pH, body temperature, is extremely difficult. It requires harsh chemistry that the cell just can't support. So the cycle uses a clever strategy called condensation.
This is step 1. The enzyme is citrate synthase.
Right. The enzyme takes that 2-carbon acetyl-CoA and essentially smashes it together with a 4-carbon carrier molecule that's already in the cycle. That molecule is called oxaloacetate. So 2 carbons plus 4 carbons equals 6. We get a new 6-carbon molecule called citrate.
But why make a bigger molecule if the ultimate goal is to break it down? That seems counterintuitive.
It's all about chemical activation. By attaching that stubborn 2-carbon acetyl group to the bigger oxaloacetate molecule, you completely change its chemical environment. You turn that unreactive methyl group into a methylene group that's situated next to other reactive groups. In plain English, you have to build it up first in order to make it breakable.
Yeah, there's a beautiful safety mechanism built into this enzyme, too. The idea of induced fit.
Oh, this is crucial. Acetyl-CoA contains a high-energy thioester bond. You can think of it like a loaded spring, just packed with energy. If a water molecule were to get into the active site prematurely, it could hydrolyze that bond pop, the spring, and release all that precious energy as useless heat without doing any work.
So the enzyme has to be very, very careful.
Extremely careful. Citrate synthase has a conformation that's chemically open when it's empty. It binds to the 4-carbon oxaloacetate first. Only when oxaloacetate is properly seeded does the enzyme physically clamp down. It changes shape by about 18 degrees, and that clamping motion creates the binding pocket for acetyl-CoA.
So the fuel slot doesn't even exist until the partner molecule is already strapped in.
Exactly. It's a security measure. It ensures that the high-energy acetyl-CoA is never brought into the active site unless its reaction partner, oxaloacetate, is already there waiting for it. It completely prevents the accidental waste of that thioester bond energy.
Okay, so now we have citric, a 6-carbon molecule. And here, Medlock Holmes is where we hit a fascinating historical snag, the prochiral mystery.
Ah, yes. The Ogston paradox. This is one of my favorite stories in all of biochemistry because it shows how even brilliant scientists can get completely tripped up by simple geometry.
So lay it out for us. Citrate, if you look at its structure, it's a symmetric molecule, right?
That's correct. If you build a model of citrate, it has a central carbon with a hydroxyl group and a carboxyl group attached. And then coming off that central carbon, it has 2 arms that are chemically identical, a CH2COO- group.
Identical arms. So if I hold it up to a mirror, it looks the same. If I spin it around, the left arm looks exactly like the right arm.
Chemically, in a test tube, they are completely indistinguishable. If you reacted citrate with some random chemical, that chemical would hit the left arm 50% of the time and the right arm 50% of the time. Pure chance.
When the early biochemists were tracing this pathway using radioactive labels, they found something that seemed totally impossible. The very next enzyme in the cycle, aconitase, it always worked on the same arm, 100% of the time. It was treating a perfectly symmetric molecule as if it were asymmetric.
And this caused a huge panic in the field. People said, that's impossible. An enzyme is just a chemical catalyst. It can't tell the difference between 2 identical groups. Therefore, citrate cannot be the true intermediate in this cycle. They were ready to throw out the entire Krebs cycle theory because of this one observation.
Until Alexander Ogston comes along in 1948. He saved the day with a simple, elegant thought experiment.
He pointed out something that seems obvious in retrospect, but was absolutely revolutionary at the time. He said, and I'm paraphrasing, a symmetric object can be treated asymmetrically if it is placed into an asymmetric container.
I needed the coffee mug analogy from the textbook to really get this.
It's a great analogy. Imagine a plain white coffee mug with a handle. That mug is, for the most part, symmetric. Now look at your hand. Your hand is not symmetric. You have a thumb on one side, fingers on the other. It is chiral.
Okay, got it.
Now, try to pick up the mug. If you grab it with your right hand, there's really only one comfortable, secure way for it to fit. Your thumb goes one way, your fingers wrap around the other. By placing the symmetric mug into your asymmetric hand, you force it into one specific, non-random orientation.
So the enzyme's active site is the hand.
Yes. The active site of aconitase is the hand. It's an asymmetric environment. It has 3 specific binding points, let's call them A, B, and C, that have to interact with the citrate molecule. Because the enzyme is made of amino acids, which are themselves chiral, that binding site is inherently asymmetric. So when the symmetric citrate molecule floats in, it can only dock in one very specific way to make contact with all 3 of those points.
And because it's locked in that one single position.
Only one of those 2 chemically identical arms is actually pushed into the reaction zone of the enzyme. The other one is pointing away, out into empty space. The enzyme doesn't choose which arm to modify. The geometry of the binding forces the choice.
That is the famous 3-point attachment theory. In this context, geometry is destiny.
It is, and it was a beautiful piece of logical deduction that saved the entire theory of the Krebs cycle from being discarded.
So aconitase grabs citrate, does a little shuffle, and turns it into his isomer, isocitrate. But Leninger mentioned something else about aconitase that just blew my mind. It has a secret identity.
The moonlighting protein.
It sounds like it's driving for Uber on the weekends to make extra cash.
In a way, it is. By day, aconitase is a humble metabolic worker in the mitochondria. It has a little iron-sulfur cluster at its core that helps it do its chemistry. But the cell has a different problem it needs to solve. It constantly needs to know how much iron it has. Is the iron level high or low?
And who better to ask than an enzyme that actually uses iron as part of its structure?
Exactly. It's an incredibly elegant solution. If the cell's iron levels get too low, the iron-sulfur cluster inside the aconitase enzyme literally falls apart. The tool breaks because a key part is missing. But instead of the now useless protein being sent for recycling, it changes its shape. It refolds into a new form we call apoaconitase.
And what does this new version of the protein do?
It leaves the metabolic factory floor and travels to the cytosol. There, it functions as an iron regulatory protein, or IRP1. It binds directly to messenger RNA. Specifically, it binds to the genetic instructions for making ferritin, which is the protein that stores iron, and the transferrin receptor, which imports iron into the cell.
So when cellular iron is low, the metabolic worker transforms into a genetic regulator and sends out a clear message. Stop storing the iron we have and open the gates to import more.
Precisely. It's a beautiful direct feedback loop. The enzyme that uses the resource is also the sensor for the resource. It cuts out the middleman and makes the system incredibly responsive.
Nature is just so incredibly frugal and efficient. Okay, let's move down the assembly line. We have isocitrate. Now we are entering the energy harvest phase. We're about to chop some carbons off this molecule.
This is the first of 2 oxidative decarboxylation steps. We are going from a 6-carbon molecule down to a 5-carbon molecule. The enzyme that does this is isocitrate dehydrogenase.
And this is where we get our first real payout from the cycle.
Yes. We produce our first molecule of NADH. We've captured a pair of high-energy electrons, and we lose a carbon atom, which is released as carbon dioxide, CO2.
I want to pause on that for a second. The CO2. This is literally the CO2 that we breathe out.
Yes, absolutely. When you exhale, you are breathing out the broken carbon bonds of, well, whatever you had for breakfast. This step and the very next one are the primary sources of the CO2 that your body produces.
Okay, so we are now down to a 5-carbon molecule, alpha-ketoglutarate.
And alpha-ketoglutarate is a real VIP. It's an incredibly important branch point molecule. We'll see later that it's a major entry and exit point for many amino acids. But for the cycle to continue, we need to oxidize it again.
So we go from 5 carbons down to 4. We lose another molecule of CO2. We generate another molecule of NADH. And the enzyme here is the alpha-ketoglutarate dehydrogenase complex.
And here is a fantastic systems thinking moment from Leninger. If you look at the structure of this enzyme, alpha-ketoglutarate dehydrogenase, it should look very, very familiar.
It looks almost exactly like the PDH complex we started this whole journey with.
It's uncanny, isn't it? It has the same multi-part E1, E2, E3 architecture. It uses the exact same 5 cofactors we talked about earlier. It even uses the same swinging Leiper arm mechanism to channel its substrate.
It's a literal copy-paste job.
It's a perfect example of evolutionary recycling. Nature figured out a highly efficient mechanism for oxidizing a particular type of molecule, a keto acid, that was the PDH complex. When it needed to evolve an enzyme to oxidize a slightly larger keto acid, it didn't reinvent the wheel. It just took the genes for PDH, duplicated them, and then tweaked the active site of the E1 component just enough to fit the larger molecule. These enzymes are what we call paralogs.
But just like the PDH complex, this step represents a major point of vulnerability. And Harper highlights a really frightening clinical situation here, hyperammonemia. This is what happens when the liver starts to fail.
The liver is the body's main waste treatment plant. One of its most critical jobs is to take toxic ammonia, which is a byproduct of amino acid breakdown, and convert it into non-toxic urea, which you can then excrete. If the liver is badly damaged, say, from cirrhosis or hepatitis, ammonia builds up in the blood. And that ammonia can cross the blood-brain barrier.
And the brain, I assume, tries to protect itself.
The brain does its best to detoxify itself. It has an enzyme that can attach a molecule of ammonia onto alpha-ketoglutarate to produce the amino acid glutamate. It's essentially using the alpha-ketoglutarate from the Krebs cycle like a sponge to soak up the toxic ammonia.
But alpha-ketoglutarate is the fuel for the next step of the Krebs cycle.
Exactly. By using it as an ammonia sponge, the brain rapidly depletes the concentration of alpha-ketoglutarate inside its mitochondria. The entire citric acid cycle stalls because a key intermediate has been siphoned off.
So the engine stops because someone stole the spark plugs to use them as paperweights.
That's a pretty good way to put it. The result is that ATP production in the brain plummets. This leads to the neurological symptoms we see in hepatic encephalopathy, coma, seizures, death. It's a stark illustration of how the cycle's function depends on maintaining the concentrations of these intermediates. If you pull one block out of the Jenga tower, the whole thing comes crashing down.
So assuming our liver is working properly, we've successfully gone from 5 carbons to 4. We are now at a molecule called succinyl-CoA.
And succinyl-CoA, just like acetyl-CoA, has that high-energy thioester bond. It's another one of those loaded springs. The cell is now going to use the energy of breaking this bond to do something unique in the cycle.
This is the only step that involves substrate-level phosphorylation.
Right. Most of the ATP we get from our food comes later, indirectly from the electron transport chain. But here, the energy release from breaking that thioester bond is so large that the enzyme succinyl-CoA synthetase can use it to directly slam a phosphate group onto a GDP or an ADP molecule.
It makes a molecule of GTP or ATP right there on the spot.
Right on the spot. It's like getting cash in hand versus a check that you have to take to the bank later. It's a small but direct energy payoff.
Okay, so now we have succinate, a 4-carbon molecule. We've done all our decarboxylations. The goal now is to regenerate our starting carrier, oxaloacetate. This involves 3 more steps, and the first one, involving succinate dehydrogenase, is a bit of an oddball. It's not floating in the matrix like all the others.
No, it's the only one that's different. Succinate dehydrogenase is physically embedded in the inner mitochondrial membrane. It's actually a dual-purpose enzyme. It's part of the citric acid cycle, but it is also complex 2 of the electron transport chain.
So it's double dipping. It has a foot in both pathways.
Yes. It takes the electrons from succinate, but instead of putting them on NAD, it puts them onto a different carrier, FAD, to make FADH2. And it feeds those electrons directly into the electron transport system. It is the physical, tangible link between the cycle and the final stages of energy production.
There's a cool little detective story here involving a poison called malonate.
Malonate, a classic competitive inhibitor. It's a molecule that looks almost exactly like the real substrate, succinate, but it has 1 less carbon in its backbone. So it fits perfectly into the active site of the enzyme, but the chemistry can't happen. It just gets stuck.
It jams the lock without turning the key.
It jams the lock. In the early days of biochemistry, before they had worked out the whole sequence, scientists used malonate as a tool. They would poison a cell preparation with methanate and then just see which chemical piled up behind the roadblock. And what piled up was succinate. That's how they knew that succinate was the molecule that came immediately before the step catalyzed by succinate dehydrogenase. It was forensic accounting with molecules.
That's clever. So succinate becomes fumarate. The next enzyme, fumarase, adds a water molecule to make malate. And finally, the last step, malate dehydrogenase oxidizes malate back into our starting material, oxaloacetate. We're home.
But this last step is thermodynamically fascinating. If you look at the free energy change, the delta G for this reaction under standard conditions, it's actually positive, very positive.
Positive. Wait, that means it's an uphill reaction. It shouldn't happen spontaneously. The reaction should actually run backward.
Chemically, it wants to run backward. The equilibrium lies far in the direction of malate, not oxaloacetate. So the question is, how on earth does the cycle ever finish? Why doesn't it just get stuck at malate?
I feel like there's a trick coming. There has to be.
The trick is to look back at step 1. Remember citrate synthase.
The enzyme with the induced fit, the one that starts the whole cycle.
And we talked about how energetically favorable that reaction is. It has a hugely negative delta G. It's like a massive waterfall. Because it's so energetically favorable, it acts like a powerful vacuum cleaner. It instantly sucks up any molecule of oxaloacetate the moment it gets made by malate dehydrogenase.
So the concentration of the product, oxaloacetate, is always kept at almost 0.
Practically 0. And by Le Chatelier's principle, by constantly removing the product, the cell pulls that thermodynamically unfavorable uphill malate reaction forward. The massive push from the first step of the cycle is what pulls the difficult last step across the finish line.
That is just elegant. It's chemical teamwork across the entire pathway. So, we have closed the loop. We're back to oxaloacetate, ready for another turn. But both Leninger and Harper really stress that we need to stop thinking of this as just a simple closed loop.
Right. We absolutely have to talk about the amphibolic nature of the cycle.
Amphibolic. It sounds a bit like amphibious.
It comes from the same Greek root, amphi, which means both. The cycle serves both catabolic purposes, that is, breaking things down for energy, and anabolic purposes, which is building things up.
This brings us back to the traffic circle or roundabout analogy you mentioned earlier.
It's the best way to think about it. Acetyl-CoA is the car entering the roundabout. But cars don't just drive in circles forever. They take exits to get to different destinations. The Krebs cycle has multiple exits.
What kind of exits are we talking about? Where do they lead?
Well, for example, citric can be pulled out of the mitochondria and used in the cytosol as the starting material to make fatty acids and cholesterol. Alpha-ketoglutarate can be siphoned off to become the amino acid glutamate, and from there, other amino acids. Succinyl-CoA can leave the cycle to become the precursor for the heme group in your hemoglobin.
So your blood, your muscles, your fat storage, they're all constantly drawing raw materials out of this central cycle. It's a supply depot. But here is the problem that Medlock Holmes spots immediately. If everyone keeps taking an exit, Eventually, the traffic circle is going to be empty.
And that's the problem of cataplerosis, literally emptying down. If you drain all the alpha-ketoglutarate to build muscle protein, you'll have nothing left to turn into succinyl-CoA, and eventually nothing to turn back into oxaloacetate. If you run out of oxaloacetate, you can't pick up the next molecule of acetyl-CoA that's waiting to enter the cycle. The entire engine stalls.
You need a way to refill the tank.
You need anaplerosis, from the Greek for filling up.
Yeah.
You need reactions that can replenish the cycle's intermediates.
And the most important anaplerotic, or refilling reaction, involves an enzyme called pyruvate carboxylase.
This is the emergency tanker truck. Normally, as we saw, pyruvate from glycolysis gets turned into the 2-carbon acetyl-CoA to enter the cycle as fuel. But pyruvate carboxylase can take that same 3-carbon pyruvate and, in one step, turn it directly into the 4-carbon oxaloacetate.
So it completely skips the start of the cycle and just dumps a finished intermediate right back into the middle.
It bypasses the acetyl-CoA step entirely. It's a shortcut that dumps 4 carbon units straight into the mix to repressurize the system and get traffic flowing again.
And Leninger shows us the mechanism here involves our old friend biotin, another B vitamin.
Biotin, yes. Just like the Lipotor arm in PDH, biotin is attached to the enzyme by a long, flexible tether. Its job is to carry CO2. It picks up a CO2 molecule at one active site on the enzyme, and then its long arm swings that CO2 over to a second active site to stamp it onto pyruvate, turning a 3-carbon molecule into a 4-carbon one.
And Harper adds a really critical regulation detail here. Pyruvate carboxylase is allosterically activated by acetyl-CoA. It requires acetyl-CoA to work. Why is that important?
Think about the logic. When do you need to refill the cycle with oxaloacetate?
Well, when the cycle is running low on it.
Yes, but more specifically, you need to refill it when you have lots of fuel, acetyl-CoA, waiting to enter, but not enough of the carrier molecule, oxaloacetate, to pick it up.
So the backlog of fuel is the signal that turns on the pump to add more carrier.
Exactly. A high concentration of acetyl-CoA is a direct signal to the cell that says, hey, we have a traffic jam at the entrance. We've got plenty of fuel, but not enough buses to put it on. Turn on the pyruvate carboxylase and make more buses. It's a perfect supply and demand feedback loop. It ensures the cycle is only refilled when there's actually fuel waiting to be burned.
That brings us really neatly to the broader topic of regulation, the control knobs for the whole system. Because the cell doesn't want this cycle running at full blast when you're, asleep.
The primary regulators are, as you'd expect, the energy status of the cell. The cell is constantly measuring its own battery charge.
The ATP to ADP ratio.
Exactly. If you have high levels of ATP and high levels of NADH, that's the signal that says we are fully charged, we have enough energy. These molecules act as allosteric inhibitors. They bind to the key regulatory enzymes PDH, citrate synthase, isocitrate dehydrogenase, and they slow them down.
And conversely, if you have high levels of ADP or AMP,
that's the low battery warning. Those molecules signal a need for energy, so they act as activators and speed the enzymes up. But in certain tissues, like muscle, there's a special gas pedal, calcium ions.
Calcium. Usually I just think of calcium in the context of bones or maybe nerve signals.
In physiology, calcium is the universal trigger for muscle contraction. When your brain sends a signal to your bicep to contract, the nerve releases a signal that floods the muscle cell with calcium.
Right, that's the final step that makes the muscle fibers slide past each other.
But that same flood of calcium also rushes into the mitochondria inside that muscle cell, and it directly stimulates the dehydrogenase enzymes of the Krebs cycle.
So the signal to move the muscle is also the signal to make more energy to power that movement.
And it does it instantly. It's what we call a feed-forward mechanism. It doesn't wait for the ATP levels to drop first. It revs the engine the very moment you step on the gas pedal. It anticipates the future need for power.
That is just incredibly responsive design. Now, we have to talk about when this intricate regulation goes wrong. The dark side of metabolism. We've touched on vitamin deficiencies and poisons, but let's talk about something more systemic. Cancer.
This is a massive and relatively new area of research. For a long, long time, we thought cancer was just about genetic mutations and growth factors and tumor suppressors. But we now know that fundamentally altered metabolism is a core hallmark of cancer.
And both Harper and Leninger mention a concept called oncompatibilites, specifically mutations in the enzyme isocitrate dehydrogenase, or IDH.
This is a mutation that is incredibly common in certain types of brain tumors, like gliomas. The mutation breaks the enzyme. So instead of making alpha-ketoglutarate, it accidentally makes a different, weird, useless-looking molecule called 2-hydroxyglutarate.
And what does this weird molecule do? Is it toxic?
It's not directly toxic. It acts as a mimic. It looks just enough like alpha-ketoglutarate that it can bind to a whole class of other enzymes that normally use alpha-ketoglutarate as a cofactor.
Yeah.
But it jams them. Specifically, it inhibits enzymes that regulate the structure of your DNA enzymes called histone demethylases.
Hold on. So a broken metabolic enzyme produces a rogue metabolite that ends up changing the genetic programming of the cell.
Yes. It fundamentally alters the cell's epigenetics. It silences tumor suppressor genes and locks the cell in a state where it can't differentiate properly. It stays in a primitive, rapidly dividing state. It's a direct causal link from a single error in the Krebs cycle to the genetic dysregulation that drives cancer.
And that's separate from the more famous Warburg effect.
Right. Otto Warburg noticed this almost a century ago. Many cancer cells seem to shut down their mitochondria and rely almost exclusively on glycolysis, on fermentation, even when there is plenty of oxygen available.
Which seems completely backward, doesn't it? Glycolysis produces way, way less ATP than the Krebs cycle and oxidative phosphorylation. Why would a rapidly growing tumor choose the inefficient energy path?
Because cancer cells don't just need energy, they need stuff. They are biomass production factories. They need to divide constantly, which means they need to make new cell membranes, new DNA, new proteins at an incredible rate.
So they deliberately shut the door to the Krebs cycle to create a resource backup.
That's a leading theory. They create a bottleneck so that the intermediates of glycolysis, like glucose 6-phosphate and pyruvate, pile up. Those intermediates can then be diverted out of energy production and into biosynthetic pathways. They sacrifice energy efficiency for material abundance. They choose to use the carbon atoms to build the house rather than burning them in the fireplace for warmth.
That is a fascinating and chilling strategic trade-off. Before we wrap up, I just want to mention one last poison because the mechanism is just so devious. Fluoroacetate.
The ultimate Trojan horse poison.
It's found naturally in some poisonous plants in Australia and Africa. It was also used as a potent rat poison.
The cell is completely fooled by it. The fluoroacetate molecule looks almost identical to regular acetate. It just has one hydrogen atom replaced by a fluorine atom. So the cell's enzymes pick it up, activate it to fluoroacetyl-CoA, and then citrate synthase grabs it and combines it with oxaloacetate to make fluorocitrate. To the cell, it's all just business as usual.
Until that fluorocitrate gets to the next enzyme, aconitase.
Right. Fluorocitrate enters the active site of aconitase. The enzyme tries to do its usual chemical reaction, but that single, tiny fluorine atom gets in the way and locks the mechanism solid. It forms an unbreakable bond with the enzyme.
It's a suicide substrate.
The enzyme literally kills itself by trying to process the poison. And because every molecule of aconitase is now irreversibly blocked, citrate piles up, the entire cycle grinds to a halt, and cellular respiration ceases. Game over.
All because of one tiny atom being in the wrong place.
It just goes to show how exquisitely precise the geometry and chemistry of these enzyme active sites really are.
Wow. We have covered a massive amount of ground today. From the giant PDH machine and its swinging robotic arms, to the moonlighting enzymes, the traffic circle logic, and the deadly precision of these poisons.
It really validates that metabolic crossroads byline, doesn't it? It touches everything.
So as we've synthesized all this, Laninger's elegant logic and Harper's clinical medicine, what is the one final image you want to leave the listener with? The one thing to change how they see this pathway in the textbook?
I want them to update their mental image of the mitochondrial matrix. We often think of these enzymes, citrate synthase, aconitase, and so on, as just floating around randomly in a watery soup, bumping into things by chance. But the emerging evidence points to something far more organized, a concept called the metabolon.
The metabolon.
It's the idea that these enzymes, which are sequential in the pathway, might not be floating free at all. They might be physically clustered together in a non-covalent super complex, a kind of solid-state factory.
So the intermediates aren't even really floating freely between enzymes. They're being passed directly, channeled from one active site to the next, just like we saw with the PDH complex at the very beginning.
Precisely. The soup of the matrix might actually be a highly organized, almost crystalline factory floor. We are just beginning to develop the techniques to see the true structural organization of these pathways in living cells. It's not just chemistry, it's architecture.
So the 2D diagram in the textbook is just the circuit diagram, the schematic, but the reality is 3D, dynamic, and breathtakingly integrated.
Exactly. And that integration, that architectural elegance, is what allows life to exist at the speed, efficiency, and complexity that it does.
Medlock Holmes. I think our case is closed for today. This has been a fascinating look into the engine room of the cell.
It was a pleasure to investigate with you.
And to all of you listening, next time you take a breath and exhale, remember where that CO2 is coming from. Remember that microscopic factory floor spinning away inside every one of your cells, stripping electrons, refilling the tank, and keeping the lights on. It's a very busy world down there. We'll see you in the next deep dive.